Botanical characteristics of taro. Skull. Mono skull. Colocasia esculenta is an herbaceous plant, usually from 0.5 to 2.0 meters tall. The mon tree. The skull consists of a central bulb usually lying on the ground, from which the leaves grow upwards, roots grow downwards, while tubers, cormels, armpits, daughter corms, and beef bands, stolons, grow horizontally to the sides. Roots. The root of the mon species. The skull root is the root of the root that grows from the germination around the bulb. Short roots, horizontal direction, and growing into layers in an upward direction with the development of burning. Tubers. The roots are usually white and contain anthocyan. Some genotypes have two types of roots at the same time, pigmented and pigmented roots. The roots grow into many layers, depending on the number of leaves of the plant. The number of roots and the length of roots depends on each variety and soil. An average root layer has from 25 to 30 roots. Root crop research team, 1969. Tuber. In both taro and potato forms, the skull has a root bulge or tuber stem containing starch. The main stem is considered to be the main stem structure of the plant, located in the soil. On the tubers, there are many burns, each with growth sprouts. After the leaves are gone, add a burning stem and long stem. The root surface is marked by a circle called the longitudinal leg. That is the connecting point of the scales or old leaves. Many lateral sprouts are distributed on root burns. The top of the root is the growth point of the tree. The sprouting of plants all starts at the top of the bulb. The whole vertical part of the leaves on the ground creates a false body of mon and skull trees. Potato tubers. Skulls are very different in size and shape depending on genotype, tuber types, and ecological factors, especially those that affect the tubers such as structure and structure of soil. The presence of stones. The bulbs of representative varieties grown on high land are usually round or slightly long, while the long-rooted varieties are usually those grown in water fields and marshes, ditches, ponds. All females, tubers and armpits have an almost identical outer structure, each with a spike at the apex and many axillary spores of countless scales on the stem. In the varieties of taro, tubers can reach a length of about 30 centimeters and have a diameter of about 15 centimeters.
But the tubers of the potato varieties are usually smaller. Both female tubers and tubers are composed of three parts: outer skin, shell, and tuber meat. The outer shell may be smooth,
grainy, or covered with scales that are usually dark brown in color. The mantle is between the outer shell and the core. The jacket and tuber core consist mainly of parenchyma. In the outer core of the cell contains a lot of starch granules also have tubers. The amount of tuber is very different between genotypes and is influenced by the environment. Pigments in tubers range from white, light yellow, dark yellow, orange to pink, red and purple red. In addition, in some varieties of colored flesh intercalated between white and purple red or purple ores white tissue with darker fibers. Occasionally in the field observed in some varieties with cow strips. Horizontal development on the surface of the soil. From the eyes of the strip, the roots will grow and the buds grow into new ones. Leaves. The main leaf is the only part seen on the ground. The leaves determine the height of the potato. The leaves of mon and skull trees are relatively large. Each leaf is composed of a straight leaf stalk and a leaf blade. The leaf blade of most genotypes is shield-shaped, heart-shaped, with the umbilical cord near the middle, which is the point between the stem and the leaf blade. The leaf blade is smooth. The length can vary from 20 to 70 centimeters and width from 15 centimeters to 70 centimeters. The size of leaves is greatly influenced by external conditions. Taro leaves, skull, are the largest in the flowering period. Leaf blade color ranges from light blue to dark purple depending on genotype. Leaves can indicate one color or add other spots or streaks. Taro leaves, skulls can also be discolored when sick, especially when infected. On the leaf blade there are three main veins, a straight rib from the point of connection along the leaf to the leaf blade to the top of the leaf blade. The other two tendons run horizontally to the two apical lobes. From the three main tendons, there are many small, emerging veins forming a mesh. Petiole in taro. Skull is often called leaf. In the parenchyma of vertical leaves, there are many areas that look along the taro leaves. Skull is often porous. Along the fat leaves there are sheaths that cling tightly to the root to form the old body. Leaf length varies depending on genotype from 25 cm to 160 cm. Vertical leaf color ranges from yellow-green to dark purple, sometimes with purple or dark blue stripes. Vertical and leaves are not always the same color. The sheath of the vertical is usually a hug that is about 1/3 of the length of the vertical. Nearly the harvest of tubers, the leaves are getting shorter and shorter and the leaves are smaller. Flowers, fruits and seeds. Flowers of the montree, the monogamous flowers, skull, male and female flowers on the same axis. The inflorescences are in the form of cotton, growing from the tubers, shorter than the petioles. Each tree can have one or more flower clusters. The inflorescence is made up of a short stalk, a flower axis and a bag. Flower stalks are green to yellow or purple depending on the variety. The structure of flower stalks is similar to that of vertical leaves. The bag has two parts. The upper part is yellow. The lower part is green. The length is about 20 centimeters and the flower axis is held. Flower axis is shorter than has 4 parts. The bottom of the flower, followed by a non-reproductive part. The upper part is the male part, and the last part is the non-reproductive, spiky appendage. Flowers do not have covers. Male yellow flowers have multi-edge bivalent, round pollen grains, anther chalk. Female flowers have one box, very short hose. Berries are about 3 to 5 centimeters in diameter and contain many seeds. Each non-embryo particle has an endosperm. Taro skull is a highly variable species. This has been mentioned by many researchers, Ivancic and Labat 1999. So far, it has not been possible to describe all of its morphological variations. In most cases, the scientists only describe the most important and stable genetic traits. Like other crops, in the taro skull, the new genetic variants often export. It is currently the result of natural and recombinant mutations. Classify taro. Sweet potato skull of the genus Colocasia is one of the most important genera in the family, Herbaceae. Due to the long history of clonal propagation, there is still a lot of uncertainty about the problem of vegetation classification of this genus. The varieties of taro cultivated skull are classified as Colocasia esculenta, a polymorphic species. The species in this genus are used as food, human food and animal feed. The genus Colocasia was determined by Schott in 1832 based on the two species described by Linnaeus for the first time in 1753 as Arum Colocasia and Arum esculentum, Hill, 1952. Schott also renamed the two species Colocasia esculenta and Colocasia antiquorum. Currently, there is still a lot of controversy in the Colocasia classification research. Some botanists classify that there is a polymorphic species, C. esculenta and, to a lesser extent, known as C. esculenta var esculenta and C. esculenta var antiquorum, Ghani.1984. Others argue that the genus Colocasia has a polymorphic species of C. antiquorum and at the subspecific level of C. antiquorum var typica, C. antiquorum var euchlora, C. autoquorum var esculenta, Kumazawa et al. 1956. However, there are schools that say there are certainly two species of C. esculenta and C. antiquorum are distinguished based on the morphological characteristics of flowers. Persglove 1972. According to this view, Colocasia esculenta has the asexual appendage at the apex of the flower, which is shorter than the flower cluster. An antiquorum Colocasia species have asexual appendages at the apex of the cotton spikes that do not protrude out of the flower, longer than the male inflorescence. Currently, there are countless mon and skull varieties in the world with many plant deformations. However, most varieties belong to two main groups, subspecies C. esculenta, L. Schott var esculenta accurately describes the tree as having a large cylindrical root and very few tubers, often referred to as dishing form. According to the subspecies classification, there are two groups of plants: the group of water potatoes, subject to flooding, and the group of taro, using the roots and growing on highland. These two groups use female roots to eat, tubers to breed, and vertical leaves used for breeding. Flowers have a shorter asexual appendage than the male inflorescence. Most of these subspecies have chromosomes 2n-28, often called diploid or diploid. Subspecies C. esculenta L. shot var. Antiquorum is distinguished as having a small spherical bulb with large-sized tubers growing from female tubers, often called edo. This subspecies is mainly a group of skull potatoes. The sweet potato group is widely distributed and can be grown on paddy fields or irrigated flat land, even on sloping land using sky water. Flowers have a longer asexual appendage than the male flower part. Most of these subvarieties have a chromosome set of 2n=42, often called triploid. There is also an intermediary group with many intermediate characteristics between the two groups mentioned above. Therefore, we should call the mon and skull groups the most accurate, even if there is a polymorphism of C. antiquorum and C. antiquorum var. TV pica, C. antiquorum var. euchlora and C. autoquorum var. esculenta. Currently there are thousands of taro varieties. Skulls are being grown all over the world. The varieties are distinguished mainly by the characteristics of the bulb or the characteristics of the broom, or on the basis of agronomic or cooking quality characteristics.